Hi guys, Miss Francis here. Today we're going to finish up our DNA, the molecule of life unit. I would like to discuss how protein synthesis is similar and different in prokaryotes versus eukaryotes. So although bacteria, which are prokaryotes, and eukaryotes carry out transcription and translation in similar ways, they do have some differences. For example, eukaryotic RNA polymerases, which are enzymes, differ from those of prokaryotes and require transcription factors. Prokaryotic RNA does not get edited before translation, whereas eukaryotic RNA does. We get that tail and we get that cap as protection, and we also splice out the introns. Furthermore, their ribosomes are different. Prokaryotes have 70S ribosomes, and eukaryotes like us have 80S ribosomes. A big difference is that prokaryotes can transcribe and translate the same gene simultaneously because prokaryotes have a nucleoid region. They don't have a nucleus with a nuclear pore that they have to travel through. So they can do transcription and translation of the same gene concurrently. Whereas in eukaryotes, we have a nuclear envelope that segregates or separates transcription from translation. Now, unfortunately, there are changes that can occur in our DNA that affect protein structures and functions, and these changes are called mutations. So a mutation is a change in the genetic material of a cell or of a virus, and there are different types of mutations. If I have a chemical change in just one base pair of a gene, that's called a point mutation. Point mutations are what cause sickle cell disease, where I have a change in a single nucleotide. So instead of this T, I've got this A, and that results in producing an abnormal amino acid. Just one different nucleotide gives them one different amino acid, which causes this protein to fold and function improperly, giving this person sickle cell anemia. Now, a point mutation that results in replacement of a pair of complementary nucleotides with another nucleotide pair is called a base pair substitution. So we know that our base pair ruling is C matches with G and A matches with U when we're talking about translation. So for example, if I were to replace a G with an A, that would be a base pair substitution, right? If I were to replace a G with an A, I'm taking one nucleotide from one base pair, one complementary base pair, and replacing a nucleotide from the other complementary base pair. There are different types of base pair substitutions. We've got silent mutations where the changing of the nucleotide still causes or still indicates or still codes for the same amino acid, and in that case, there is no change in protein structure or function. However, there are other base pair substitutions that do cause detectable changes in a protein, such as missense mutations and nonsense mutations. So a missense mutation is one that I'm still coding for an amino acid, but I'm coding for an incorrect amino acid. A nonsense mutation is when the change in amino acid, when I change an amino acid codon into a stop codon. And in this case, it nearly always leads to a non-functional protein. So let me show you what I mean. Here is our wild type messenger RNA. So this is what the sequence of amino acids is supposed to look like. If I have a U inserted instead of a C, so C normally pairs with G, U normally pairs with A, so I am replacing that C with a nucleotide from the other base pair that are complementary to one another. But I am not having any effect on the amino acid sequence, then there's no change in the protein structure or function. I still had a base pair substitution, but because we know that more than one combination of our nucleotides can code for the same amino acid, sometimes base pair substitutions result in no ultimate change in the amino acids. However, in missense and nonsense mutations, that is a different story. So here I get an A instead of a G, and now instead of coding for the amino acid glycine, I'm coding for a different amino acid. So I'm still coding for an amino acid, it's just the wrong amino acid. And now the correct sequence of amino acids for that protein has been changed. Nonsense, base pair substitution mutations cause a stop codon to be created. And now none of these other codons are coded for. And that almost always leads to some sort of non-functional protein. Insertions and deletions are either I insert, I add a nucleotide pair, or I delete, I take one away. And that has a disastrous effect on the resulting protein. Unless these mutations occur in multiples of 3, because remember, it takes 3 nucleotides to create a codon, they cause what's called a frame shift mutation, where the nucleotides that are downstream of that deletion or that insertion will be improperly grouped into codons. So let me show you what I mean. Here again, we have our wild type, and this is the normal order of the amino acids. However, if I delete one nucleotide, do you see how now this codon that was once UUA is now UUG? And instead of coding for phenylalanine, I'm coding for leucine. Do you see how this is supposed to be GCC, but now it's GCU, which is coding for alanine, and so on and so forth. Now, every single codon moving down that messenger RNA is going to be different. That causes missense, because I'm still coding for amino acids, it's just that I'm coding for the wrong amino acid. Here's a frame shift causing nonsense. If I add in a U, now I have made my stop codon. Now none of this messenger RNA is going to get translated. Lastly, if I add 3 nucleotides or I take away, they're missing 3 nucleotides, I don't have frame shift. What I have is an extra or a missing amino acid. So how do these mutations occur? Well, they can occur during DNA replication, they can occur during DNA repair, they can occur during DNA recombination, and they're known as spontaneous mutations. We also know that we are exposed to mutagenic agents. These mutagens are chemicals or physical agents that cause mutations in DNA. For example, X-rays, ultraviolet light, chemicals such as asbestos. That's why when you go to the dentist to get your teeth X-rayed, he puts on that protective shield so that you're not exposing your body to these mutagens. It's what could potentially cause cancer. It's how Rosalind Franklin got ovarian cancer from being exposed to these X-rays when she was doing her X-ray crystallography. So we have reached the end of our packet. It is the end of our information on DNA, the molecule of life. The next unit that we're going to tackle involves something called DNA technology, which is a really cool unit because in current time, in the news, you will see examples of how DNA technology is helping humans. As far as the unit on DNA, the molecule of life, if you have any questions, please come see me. Please ask me. DNA replication, transcription, translation, they all have a lot of overlap, but they're also very different processes. And it's hard to wrap your head around all of these different processes. So rewatch the videos, come in and see me. We can do some practice questions, but let me know if you have any questions.